I welcome you all for today's talk. And this talk is largely on understanding 3D structure of a single molecule by Dr. Gary Wren. And he is a top scientist at Lawrence Berkeley National Laboratory. And I'll just talk a little bit about him and his work. And he works at Berkeley National Laboratory, and he holds a degree in theoretical physics and PhD in material science. And he has a postdoc training from Scripps Research Institute and Beers College of Medicine. Uh, he largely works on individual molecule electron tomography, which is related to electron microscopy as well, where you can get high resolution. And in 2000, he joined Lawrence Berkeley National Laboratory in 2006, which is a part of UCSF. And Dr. Wren has a very productive group, which we can see, which you can understand from his publications. He is being funded by one of the richest funding agencies, that is National Institute of Health. And he has more than 100 publications now. Most of them are in, a large fraction of them are in Nature, which is supposed to be the best journal in science community. Additionally, he's associate director of CAS from 2015, and I'm looking forward for a very, very, informative and very great talk by Dr. Gang. And it's wonderful to see him again. I met him in a Gordon conference, and he showed me some beautiful images, which was very, very impressive, I would say. And he's going to show some more. So, Dr. Gang, please go ahead.
Okay. First of all, thank you, Pasa, and for the great opportunity and to talk with your scientist and student. So it was very happy. And during the Golden Conference, I chatted with Pasa about important, interesting science and common interesting. So I'm very happy and have opportunity and discussion with you about my research project. So, and as Pasa introduced, I was studied material science before, and lately I go to the Scripps Research Institute for biology research. So my first PI is a Indian professor and named Alok Mitra. He's a mentor in India. Is very famous, Rama channel, and this make a structure, a structure map, a Rama channel map. So I'm very proud of him and learn a cryo-EM technology, start my biology research work at the Scripps Research Institute. So now I kind of share your my research work at the Lawrence Berkeley National Lab. And we are major focus on single molecular 3D structure. Here we don't use any average, so we do individual one. So a little bit advertisements and introduce was a molecular foundry. Molecular foundry is one department in Lawrence Berkeley National Lab, and which just beside UC Berkeley in San Francisco Bay Area. And we have like a combination science building, and they have a nanofabrication device, near field microscope, chemical synthesis, and the TEM as well. So and the department energy support this institute for combine different science and scientists to work in the same building. Therefore, they can share idea, share technology, help each other, and develop a new technology. So and we are one of the focus on electron microscope technology development. So also this is an open house in the institute, and every year we have twice in open user. So that means user come here to experiment without pay anything. You get support by scientists, by new technology, and use the expert experience. So next coming cycle will be September, very close, and So every year I have twice in the March and the September. So if somehow interesting, welcome to the webpage. Take a look at different science research direction and some technology may be useful for you. And welcome to submit the user proposal. We're open door for collaborate with people. So in the particular development, we are focused on PM is we try to see, can we see molecular in motion? So we know life in motion. And the fact is all the living stuff is not static. It's rather continually changing and growing and evolving. So as you can see here, and the dolphin in the ocean, they swim, and the bacterial cell is inactive, and cell division. Maybe I can refresh this movie. So everything is kind of in moving. However, when you go to the protein side, and we see all the protein like a rigid body, and just like a Statue of the Liberty, and they're stick there and without doing any job, and without move anything. That's kind of something gap and causing and the imaging of the protein and lost the function of the movie by experimental. So that's a technical issue because in history, to get a high resolution protein structure, people have to go to crystal in the crystal stage, and they have to lock protein in a particular position in a fast state, and therefore they got a 3D high resolution structure, but they lost the moving information. So recent cryo-EM, and they frozen sample without go crystallization. So the technology is they frozen sample in the virtual ice. Here is a small ice hole. So the white one here is a dry hole without ice. So the green one is there's an ice hole. And within the ice, and you can see the many protein here, and they are random orientation. So the cryo-EM is try to develop a technology. They from this kind of noisy image find the worst particle of the protein. For example, here's one, here's another one, and there one. Therefore, they pick up those kind of particle and use a computational method to align to average them. And after they choosing some particle have a same orientation, same conformation, and they put them together, average them together. Therefore, signal go up and noise it go down. That's why they got one view of the particle. And go another class, they find another view of particle. They have to select some particle have a similar orientation, put them together. So this is the method, and a very success. You can get a high resolution structure on some particular domain. For example, this transmembrane domain structure is so good. And however, the structure also have problems somewhere, and due to flexibility. And when they average together, and the structure completely disappear by average from different molecule. They have different conformation. So this already breakthrough and got a Nobel Prize in 2017. And due to this new method without go crystallization, and still got a high resolution structure. So this is a beautiful high resolution progress. But here you notice when they select the same particle in the virtual ice, the only like 10% data is selected. All 90% data is dropped because they don't go to the same average. So, and we know the solution is moving, and there's a big brown emotion, molecular vibration over there. And molecular dynamic simulation already proved, in the liquid stage, molecule is in a vibration due to the Brownian motion, self-thermodynamics. And in order to get molecular dynamics, there's two ways. One way, you're tracking one molecule at a different time. Therefore, you can get a snapshot at different time and put all the images in the same picture, and you figure out where's the dynamics. So another way, and the thermodynamic they already know, and you can frozen a sample in the same times by tracking different molecule. and to see what's the structure different each other. Therefore, you can recover this kind of dynamic information. So one example is NMR. They do very good job, and they choosing the sample. They choosing and structure and fit and model. You can see there come with many structure, have a different structure, but because the structure is so complex, the structure they develop is very similar each other, not large scale, quite different, and that's kind of limited. And now we think, can we do one molecule? And when the sample freezing, we don't use average, just go one by one. We don't give up any one, just choosing every single one and get a 3D map. So the first requirement is you must be see the protein. And this is kind of transmission electron microscope, give those kind of imaging. And electron tomography is the same as a hospital CT. And we can tilt the sample and have different angle. And just like a hospital CT, tilting an image different direction about the protein from different sample, and that got a 3D structure. So the same logical we have here, and we use a electron microscope. We tilting the sample and have a different angle, and visualize the same protein from different angle. Therefore, we got a series of the image from different angle. So those image is a zooming image, also have different angle. And the computational, we're going to align those image together, therefore build a 3D map. So the logical same, very similar each other. Yeah, similar as a CT, but technically it's kind of much challenging because the image we had to zoom in this one. And another electron microscope, they have huge intensity of the electron beam. The energy is so high, they can quickly destroy sample. And very quickly, for example, here you can see the visual eyes is a mile away and the electron microscope. And another is the intensity so strong. And another is the sample is so small. And you see the ratio between 10 nanometer protein compared with a microscope is the same as a panic against the Earth. So that means tracking the molecular position is very difficult. When the tilting, a little vibration and the shaking, and sample go away. So, and that's the kind of super difficult text. And more important, in history, people did think whether we can do single molecule 3D. And this is the textbook and writing by Yogi Frank, who got Nobel Prize in 1917. So in his textbook, and his mentor called Walter Hope, and who is the first person in 1974 and tried to develop a single molecular cryo-EM tomography method. And he got the first one, but many people don't believe that's true because electron beam he used so strong. And they think, in this such strong beam already destroy your sample. And all you get a 3D structure is meaningless. So that's why Yaki think this is impossible. That's why we go average. We can use a low dose condition and without damage, but we average them together. But the hope is not precious for average idea because no one knows the molecule is same or not. You average them together just by try, and you could be choosing what you want. And this is the kind of big argue between Yaki and Water Hope. So we want to test at a low dose condition whether theoretically it's possible to get a useful 3D structure or not. So we do the test by add simulation data, and this is the same molecular, have different angle, and we add noise in a different level and simulate the cryo-EM image. So cryo-EM is somehow about 0.13 signal noise ratio. So, and we want to see if alignment is perfect under this noise, can we really get a 3D map, which is useful? So at a signal noise 0.25, we got a beautiful semi-Armstrong helical structure from individual molecule. And prove the theory at this noise is fine. However, if we go 0.13, and we find the noise increase significant. However, the density still possible give you like a 7-electron helix map from one individual molecule. So that means we think, okay, maybe there's no theoretical, no physical limitation. We still possible to do the work. So that's why we spend years, almost like 16 years, work on this project and try to develop various technology, handle all different problem and do it. Here's our program. And we developed this program, and this is the one example of the testing sample. No, not a real sample, and it's a simulation data. So assuming the EM imaging and object tilting at different angle, which is a very low noise level, 0.1, and the object center is a vibration, and because we take a picture, the mechanical vibration and the shift particle center. So we want to see whether we have a program which have enough capability grab this signal, align them together. So that's what we call IPET, individual particle electron tomography algorithm. So initially, you'll see if I don't align them together, the 3D is very noisy. There's almost no signal. So by gradually iteration together, we use the initial 3D as reference, align the rest of data. Therefore, the center will be aligned better. So average the aligned data, and you see the particle become better and have a shape. So if you continue to do this one, have the different size of mask, reduce noise, and the interpretation of data, you see the information gradually align together. Therefore, you can signal it, and from the reconstruction is pretty strong, and we can build a 3D map. We prove that's the kind of the software and have a capability align this noise data. So now we may take a picture is very challenging, and because we quickly tilting in any vibration around environmental, thermal vibration or mechanical vibration, even like a cable grab inside of the holder. And this is small grabbing and causing some vibration can cause the imaging move a lot and align so poorly. So, and we have to modify the instrument and also develop a software to control microscope and to target one particle at high mag without loss then during the tilting. So we have a tons of the developer. Other than that, also we provide some sample preparation method and make a high contrast imaging, and also develop a software to enhance image contrast. Also, including some missing wedge correction, because tilting we only can tilting like 120 degree instead of 180. So that means tilting range very limited. So part of them have no data at a high tilt angle. Normally they causing elongation problem. For example, the bottom one, positive and negative tilting 15 degree, you see so poorly. But with some this data is missing data is not independent. We actually can calculate from the data we have. So this is kind of example how we develop the missing wedge correction after fill the missing data on the tilting angle, so the 3D can recover very well. So this is tons of development. So I may not see everything in details. I want to show in some application what we do. So here's one example. This paper just accepted, and this is the study of the RNA self-folding process. So this process is from the DNA to RNA called transcription folding process. After finish, the molecule actually not finished. They need like a 10-hour maturation process to self-thermal vibration and go to energy state. They go to the ideal folding conformation. So this is the RNA is a custom design, human design, special nano engineer. They use this one maybe as a drug delivery vehicle and grab the drug in the middle channel and deliver it. So after folding, you see these are pretty open conformation. And 10 hours later, they become this one. And X-ray, small angle, scary, and show up this one. This collaborate with Abby and Anderson out of Denmark, and he sent sample to us after 6 hours, and they have a mixture of them. From traditional cryo-EM single party average, they only got 2 conformation. One is we call young conformation. It's a pretty open, have one helix very tilting on the surface, just like an initial state. And another is a helix almost parallel each other, not ideal as all six parallel each other, but it's kind of the turn to the mature condition. So, and this is what the traditional data, and from single particle average, they got it. And you can see, and they only pick up 9% of data and from the total image and build this 3D map. And the 9% got a mature map and still have 80% data. They don't know where it's at and what it looks like. So we first checked sample in solution and to see what's going on. This is cryo-EM, typical cryo-EM image. This is carbon and age. This is a piece of the ice. And within ice, you can see a lot of particle and in the ice like a glasses ice. So that's why you can see through it. And I pick up some of them to show. You can see there are some of it like a bundle. This is a vertical view. You can see 6 helix bundle already formed. And some is like a C shape, the bundle not yet formed. In the side view, you can see two parallel one. Some is a V shape. And talking about there, some is a sample is already folding to the ideal bundle conformation. Some is in the middle way. And you zoom in, you can see helix very well. And the side view. And naked stand as a parallel experiment. You also see the similar conformation, 6 bundle there, and a V shape, and also some open conformation and the bundle shape all exist there. That means this sample is heterogeneity. And they have different conformation and simultaneously inside of the buffer. So, and what we do is we from the sample, take a look at different angle and tracking one molecule from different angle and image them, use computer, build a 3D map from one molecule. So here's an example of the tilting image from -30, 0, +30. We have like an angle from -50 to +50. And this is the kind of example 3 image we have. And you can see this particle here and go to the 0 degree, go another one. That means you can track the molecular position. And therefore, you can pick up this kind of area. Of course, you pick up one area, you can do the 3D reconstruction by our 3D reconstruction method. Of course, you can go another one, got another one. Every single one, you can do the same procedure. So this is the example of the imaging. From one area, you can see many particles there. And we actually only focus on the central one. You want to get enough signal. That's why we use a little bit larger size, including environmental around and align the particle. Actually, we only focus on the middle one. So once align this, and you can see the noise is go down, signal go up. And gradually we can get a image of the individual particle at a different angle, build a 3D map. This is image is so small, that's why I zoom in this one to give you a better image. It's initially before we use any mask, and you can see 6 helix very clearly, vertical, and the side view, one slice, you can see helix. And after mask shapes there outside the signal, that means not cut all the information, but give you a better strong image, low pass it. And we can so clearly see the helix over there. And this is the 3D map from same corresponding angle. So, and this cross section give you so clear about the helix location. Therefore, we can fit in RNA helix into the density map. So every single helix, we can just shape and bend on this one to match EM density map. They give a different angle view, and you can see how the helix and fit inside the EM density map. So this is the resolution, something about 20 to 25 Angstrom. And although it's now high resolution, but we didn't use average, and we still got a 3D map. So we can repeat this process one by one. And for example, here we go 120 different molecular. You can see the molecular have a similar structure and like a bundle shape. But carefully look, you can see the top helix, some is very tilting and some is pretty vertically. So, and they have much wider conformation, different cross section. You can see the red dot, all 6 helix is good enough to visualize from the image. So from and give you more example in everyone how we fit in the model. So this is 3D map from 3 different angle. We can put the model inside and fit a helix and determine where's a helix 1 to helix 6, what distribution. Top 2 is more like a young conformation. Helix is pretty open, central channel is large, and the bottom 2 is more like a mature conformation. The bond are almost folding there. And this is the pretty like the mature conformation they report by single party average. But this is a much more vertical and give you a even later conformation. So, and from that one, we can got 120 different model from the image. So, what we can learn from this 120? So this is the part of analysis we begin to do. First, if we align, put all the 120 particles into the same page, and this is a different view. And the colors are from N-terminal 3' to 5' process gave different color. They could roughly align each other, but you can see all the 120 put together, the distribution much wider. It seems like an NMR structure and wide, all the particle is a vibration around center, but this structure is much wider than NMR conformation. So, and first we use we know there is a high resolution to mature and the young conformation determined by averaging. How about we want to see each particle, how they similar to the young conformation and the mature. So we use like a RMSD calculation and to align the image to see every atom position, how far from it, give the average distance. So we found that in the particle against the young, the general is a higher level. See, the small is something about 10, large is go to the almost 19. So in the 10 to 15 around. But go to mature one, you can see the small is a 5. That means so close to mature. But the distribution is kind of wide range. So that means the structure is more close to mature instead younger. And that makes sense. And because the sample we catch it is 6 hours of the maturation process. Total process is like 10 hours. So 6 hours already past half. So another analysis is we know the difference each of particle. So we name it and number it and calculate every particle, what's the difference and between their structure. So when the structure is similar in a tree map, they are close by each other. When the particles so different each other, and in this tree map, hierarchical tree map, they are pretty far each other. So they gave the beautiful tree structure, and it's kind of beautiful, but it's difficult to understand. What's a branch mean and why they have similarity in some way. You can see they are pretty wide distribution in a range. So what we know is the young conformation and mature conformation from the single particle average high resolution. There do have some particle close by, but not all of them in a very small amount. So like here, like a four against the 120, something about like a 3 or 4%, and a very small amount similar to that. But most of them quite different. And you see the longest distance between these two is also the young group, and there's a mature group. So mature group have major conformation, and the young group have less conformation. This also agree with distribution issue. So we can't learn that much, and at least we have motivation to understand. So here motivation we understand is from the folding process in the early stage to the late bundle conformation, how the structure change. So we turn the hierarchical classic map into the one-dimensional. Therefore, we can put the young conformation on end and the mature conformation other end. And therefore, they based on similarity and they reorder. And from that one, we barely see a little bit of interesting structure from the young conformation. You see particle bundle is pretty fat, open. And then go to mature conformation, they become skinny and thinner. However, and not exactly in the right order. See, here's go smaller, the smallest here, and then somehow it go up again. So this is a hierarchical, still not enough. Help us to understand what's the kind of folding process because we have difficult order. So then we go more detailed structure. Let's see about the helical orientation, pore size, how they distribute. This is kind of more detailed main bundle helix position to see what's going on, where is the pore. So from the pore size, it's a wide change distributing this size. This is a mature conformation, it's a small pore. Young conformation have a big pore and overall distribution here. Then we turn to see where's the helix 6, how much tilting. We found a major is and go small tilting angle. That means helix 6 already vertically go to the bundle, but they also have a peak. Here's a young conformation. That's why it's two peak in a high population. That's why maybe single particle average, they only pick up the average high population particle, average them together, and ignore all other conformation. So also we can do the tilting, we can do the bundle angle, we can do the bend of the helix 6, also what's the bending angle on surface, and to see the structure and whether some conformation change, for example, the bending angle, helix 6, whether have a correlation with the distance of the other helix. They show a pretty good correlation. I mean, the conformation change, some is independent, some not. For example, tilting, maybe with the distance is kind of correlated. So, we want to try to order them. We try to narrow down the some parameters correlate. So we generate 3 major conformation like angle and bending angle and size to give that called maturation percentage. So in the maturation percentage definition, and when the helical become bundle, the maturation go to the 100%. They anything far from this kind of bundle conformation, we're called young, we call less mature. So by this definition, we replot the particle structure and give this kind of maturation percentage distribution. We found that the young and the mature conformation is close to the one peak, where it's a single particle kind of 3D structure from average. So with this maturation process, this maturation percentage is similar like an energy distribution. So because it's a 3-parameter related, we cannot display 4-dimensional map from this picture, only can divide 2-parameter. How this 2-parameter against the maturation. This is like a pseudo energy map in the 3D. And every position is from the examined data. So we're fitting the data in the 3-dimensional and curvy surface. And we choosing some of them and presented a pathway that's including the young and the mature conformation into the map. You can see the particle 1714 corresponding to this particle, and 2914 in this position corresponding to this particle. So the 37, 66, all the way to 95. So from there, give it all pretty good makes sense pathway from the folding losing structure to complete folding bundle conformation. So put them for example, this pathway, of course, they have many more pathway in here. We just give example by this pathway, how the protein folding looks at bundle and the bundle one is pretty flat. There are helix 6 in the middle. And there's some bending and change of the helix bundle and causing this one curve and turn vertically go to the joint bundle. So we give the roughly conformation just like a helix 1 to 5. It's buckled by helix 6 at the beginning. So after folding, after called transcription. So interesting, they begin the folding in one helix tilting and causing this bundle bend. And after bending, they got transform as curvature. And once the curve for the certain angle, and they began stop there and waiting for helixes to join them by tilting them. So by the helixes tilting more, the curvature falling more and more at the end for a 6-helix bundle. So this is example and how we do it. And Yano can give you a more dynamic image about how our data is processed. I make this movie, and hopefully the movie can give you a more example, more dynamic image how we get a 3D map. This is the cryo-EM image. Here's the ice. Here's a carbon film. And we have a first use a machine learning denoisy, and you can see the particle contrast goes up. And what we do is just target one particle and zoom in this image. Of course, we can do any one we want. So this is just an example, one of them. So by tilting different angle, the noise is so high, and still not enough to actually see the structure very well. So this is part of way, have the reverse contrast because protein is high density one. We have the reverse contrast and do some filter and align the data more precisely and tilting again, build a 3D and use it build a 3D, the 3D and side view of the 3D, tilting view of 3D become more clearly. Roughly, barely you can see, and you can go zoom in. You can see the helix over there in the data. See 1, 2, 3, 4, 6. You see the density. In vertical view, you can see helix better. Side view is much weaker. So this is a 6-helix bundle in form. This is 3D map, and because the signal is so weak, so the map is still not high quality, and some noise are wrong, and but it's think of this individual molecule without average. That's still challenge. So this is all 3D map from 120, and we can show you how we build a model from this one. By MD simulation, we first shift the model structure into the density map and do MD simulation and energy minimize it. You can see fitting pretty good, but the density somewhere is different. For example, here, we had to shift the helix and bend the helix in order to go to the right position. So this one is an easy fit, and some is not easy. For example, this one, we are tilting a lot, especially helix 6, and in order to fit the density, but all the connection parts still allow we twist the structure, put in the right position, still possible to fit it. And this is the opposite way, and top is go narrow and bottom go open. So it's still possible to fit the helix in here. And most of them, some is a little bit disappear in density. So we don't know why. Maybe cut off the contour level is so high and didn't show up the signal, weak signal. So this is what we align all 120 particles in the same path. We assume there's following the same string in a structure folding. And we think, okay, if that way, can we put all structure in the same page by morphing from structure one to another one, give the roughly dynamic process during the folding. That's why we use the maturation percentage as a scale bar. And to see when the maturation go up, how overall structure turn to the folding conformation. So this is the kind of roughly and folding process. And not ideally, but give you a roughly picture in how the molecular vibration and folding. So this is the example we do for RNA folding. So of course we can do many things. And if the time allow, I give you a little bit more information for this nucleosome phase transition. Not only see the molecular self-folding, also the molecular interaction form a high density and aggregation conformation. We still see the pathway, how they change their molecular structure and how they kind of interact with each other, how they form an initial nucleation and then turn to high density core. So, this is a collaboration with Klaus Postman and at UC Berkeley and started this nucleosome phase transition. We know in Chrome Martin and the protein express DNA is very loosey. So in heterochromatin, and the DNA suddenly shrink together, forms very strong density and protect the DNA during the division process. So, and there are two models, one called helical fiber model. So hierarchical fabrication and the DNA wrapped together formats bundle. The bundle one form an even bigger one at the end, go to the chromatin. So, and later people found that maybe that's not right. And there may be the liquid droplet model. And the protein and form experience with a phase transition form as high density solid conformation. So in this kind of way. So we want to see how what the structure looks like. And we use a tetranucleosome example. So tetranuclear have 4 helix, four histone protein and DNA wraparound. So from the microscope and by incubation, you can see high density come out from this kind of aggregation process of the tetranucleus. So use AFM, and we also see that long time incubation, you see high density show up, and in the sample that suggests there's a phase transition process. So TEM also gave you a beautiful image, the DNA and the histone and wrap together, they become dense, form as a strong aggregation part. This is the next step in EM imaging because heavy metal may be involved, and next step not good, but resolution pretty good. And even we can not only see the histone and the wrap around, even like DNA turns, like a major groove, we still can see from EM image. So wrap around and histone, the DNA can fit the model very well from next step. But many people don't like next step because they are heavy metal involved. And even they gave high resolution, but they think structure is not native. So we had to go to a cryo-EM part. So the cryo-EM part are not easy. So the parameter effect, the transition is so much. And for example, salt concentration, they can cause conformation change. And if you have magnesium, and the conformation change much quicker. And this is 2 minutes. If you go to the temperature, increase the temperature, phase transition very aggressively, and give a long time incubation, they are also change conformation. So, and so many parameters, and including the H1 protein involved, the aggregation much quicker. This is a multi-dimensional phase transition process. It's a very difficult study. So we can't do everything. We just like focus one parameter like salt concentration to see what's going on. Salt concentration against time. So that's an example. And we have a different salt concentration. We have time. We make a 2D map. And you want to see the different stage of the conformation change. That's why we do experiment. We found that when the salt concentration low, the transition is still there, but much slower. So this data can help us to see with early stage how the chain on B structure, how they change in conformation, causing molecular interaction each other initially now. So late high salt concentration can help us zoom in to see what's high density aggregation is beginning in form as a nucleation and how they grows up. So this EM can show in our condition can have so many different morphology and different average density. You can see how the molecule actually build up together from this one. This is from next step. Next step only give you surface information, cannot fit the model, cannot get a high resolution. And what we do is go cryo-EM. We frozen the sample. First, we want proof, and the single molecular is good enough to see the DNA wrap around histone. So that's why we use a mono nucleus, and only the chain on B only have one leg on longer, one leg shorter, and we want to see whether we can see good enough higher native structure on this one. And you can see the 2 DNA wrap around in this one is clearly to see the central core and in the histone are also possible to see. So with 2 DNA sample, you see the raw data so noisy. This is very challenging as well because the DNA is so big size. That's why we kind of use a reduced ice thickness. We have to use a pretty thicker ice in order to get them free conformation in ice. That's make a contrast so poor. So, and but barely we still can see there's a DNA and there's nucleosome on different angle. And when they align together and build together, so they gave the 3D map. Those are the kind of 3D map. From 3D, we can measure the angle on the surface. and measure how much angle around plane and so much angle against the plane. So, and we can repeat this one, got different 3D structure. This 3D projection, you can see the quality pretty good, and you can see 2 layer of the DNA, and this is one particle, this is another particle, third particle. You can see every particle gave a contribution with the angle direction. So this is the example of the 30, and we have like a 47. I kind of put them together. And the mononucleosome and the 3D structure. If you align them by histone, they're so much different about DNA arm, but everything is data. Everything data, not randomly. And if we align them together, make it like a morphine, and we barely see, oh, there's some flexibility on DNA of the DNA in solution, and they have a transition EM data from one conformation to another to third one to based on hierarchical distribution. So order. So so we can get that one. Same thing we can see if I go dinucleosome, increase number of nucleosomes. Does the angle is same distribution or not? So that means we use a tetranuclear. We'll see and whether tetranuclear structure is a reflect to long DNA, long chromatin. There's tons of the nucleosome. We'll see from 1 to 2 to 3 to 4 that the angle distribution is consistent. There's only along this DNA and the history information or length make some difference. So, and we also do the tetranucleosome and also do the dinucleosome and with the tetranucleosome and to see the distribution. So the comments distribution is pretty similar. They are quite independent with how many nucleosomes on surface. Now we see increase the salt concentration. They are going to change conformation. So how they change? Increase a little bit salt concentration from original like a 5 millimolar sodium chloride to 50. So you see the nucleus, tetranucleosome suddenly shrink and become a narrow globule conformation. And this structure quite different. So if you don't use a salt and use a protein, you find that they don't shrink, but their protein is more interaction each other. So now we begin to do the statistic and to see with a one mono, how the DNA wrap around surface, where's the start, where's the end, where's the distribution. Dinucleosome, where's the start, where's the beginning, where's the end, and how the distribution. And the tetranucleosome, trinucleosome, tetranucleosome, you can see the distribution on there. And then even go the high salt and I binding. You find the I binding and change a little bit wrapping level and remove some of the wrapping level in the middle. So we'll talk it's a little bit interesting information. So also we can see where's the start, where's the end, their distribution. And also we can see what's the angle on surface, how they distribute each other by time change. You can see from mono to tetra, it's kind of similar each other, but go to high salt, the peak shift. And with H1 involved, they are not only shift, also have another peak generate from a certain position. So if you see the surface like they have a angle open and close. So they also we can do a lot of statistics. I can't tell so much details quickly go through. So they're position each other and the surface plane each other. So what do we learn from here? We learn from the salt concentration to high concentration, the surface angle changes. The angle between the DNA and the linker change a lot. Those distribution change and causing they have more chance to go cross conformation and by salt concentration change. This is maybe due to the salt concentration block the surface and causing the cross. So the conformation change just like this kind of baby toy. And when they change the angle, you see the overall size change a lot. So and this is kind of example in how we do. Of course, we can continue do the high concentration, much more conformation, much more complex one. We steer from the density map. We can feed the DNA, feed the histone, and to see the structure. So quickly give you about example and from imaging how we got a complex conformation. This is a molecular interacting each other. This is 3D map, density map from EM imaging. And this is where the software found where's the high density part. And where's the kind of another layer is the DNA portion, and from that one they begin to fit the model from this kind of aggregation conformation. So this is the background lattice support supporting field. They use a lattice support the structure. So by fitting, they begin build a model from this one. So this kind of histone and DNA wrap around overall structure. So give the morphology, and from this morphology, you can do statistic, like how much size about those kind of aggregation, how they kind of interact with each other. So and we can do too many things, different temperature. You see the conformation change, and this is how the model built up from each micrograph. So this is early stage, and later stage, you can see how everyone can fit in the map with it's still a challenge because it's aggregation. Once they form a high density, the fitting become more and more challenging. So we only roughly fit in, not precisely. And what do we learn from this one? And if we put the different aggregation component together, you can see the early stage. And they're kind of uniform distribution, and the DNA and the nucleosome become aggregate each other and become uneven distribution and form as an initial spin nodal conformation. This is kind of initial conformation form. Then once they form, it's a kind of high density one merge together, generate large aggregation conformation from the image. So, and they give the roughly morphology and how the aggregation form. You see the different density also join them together. So, this is kind of phase transition process. And the transition process was speed up a lot by H1 join. This is the protein speed up. This is the aggregation, general addition link on this one. So, and I kind of can stop and talk here. And with the individual one, we don't average everything. We become a lot of things we can study for antibody flexibility and antibody binding with drug, what's the conformation change, and how the human IDL, VLDL, and even Corey and the particle, we can get a 3D map without average, and even like a drug developer, and to see how the morphology change during the for the transfer process for cholesterol. So thank you very much for your time. Probably I run out my time, and hopefully I still have a little time to answer your question. Thank you very much for your attention.
Thank you so much, Gary. Great talk, I would say, with a lot of emerging science at molecular level, and great to see also the unpublished work, which is yet to be published, and which seems to that the work is at the frontier of science and structural biology. Uh, I can just say that the talk will be archived on YouTube for the rest of the world to catch up with the kind of research that you're doing right now. Yeah, so do any of you have any questions that you want to ask? Then we can go ahead with that. Yeah, yeah, come. Yeah. Hi, sir. Yeah, am I audible?
Good.
Okay, so actually my question was that generally when we find what is the resolution of our image with the help of this FSC curve, so it is actually what happens, it actually drops and cuts that threshold value, and then it almost saturates in a higher frequency values. But the thing is that many times in one of the two data set, what I observed, it actually it intersects that threshold line. And after that, going to the saturation value or going down, it actually comes up, and from the bottom, it cuts that this threshold value, and it start oscillating kind of in that threshold region. So is it some mistake, or if it is a mistake, then it is during the data taking process,
Sorry,
or due to
you
the
speak a
process?
little bit fast. I didn't 100% catch it. So can you a little bit slowly and talk what's the key part? Your question?
Yeah, actually, key question is that during the FSC curve, that
Okay.
correlation actually crosses that threshold value, right?
Yes.
So I observed that in one or two data set, what happened, that threshold value actually intersect that threshold value, and after that it start oscillating in that region. It is actually intersecting that threshold value again and again. It is actually kind of oscillating in that region.
Okay, probably you're talking about this one. And this is the FSC
Yeah,
from EM
it's
imaging.
perfect. It's
Yes.
perfect. Actually, it's dropping down in, but in
Oh,
one or two
they
data set
dropping
which
down.
I
They
processed, it
dropping down
was like that.
literally. They didn't come back. So yeah, the review asked the same question. That's why I update imaging, but I didn't update this one. So FSC go to here. They didn't go up. They keep
Yes.
continue low. So maybe the data I have other slides and show this one. FSC go down. They didn't go up, and they can keep consistent low in this prediction. That's a good question. So to the short script, they didn't come out. And we only zoom in the bottom one, you want to see, and their calculation between two map, half data map. If we use a tilt angle and we use a frame to separate, this is two way to separate data. One use a tilt angle, even odd data, or use a frame to separate. How the FSC and depend on methodology different. That's why we want to see show a little bit of two method different. We zoom in the FSC curve, but then we ignore and we didn't realize, people interested to see where's the FSC change after this the resolution calculation. They keep down. So I hope I can show you quickly about this one for our new data. So where's this one figure? And zoom in so I can share another PPT or recent figure. Okay, a little bit huge, so loading take a little bit time. So not this one. Sorry, or should be H6. RNA should be RNA1. So switch slice and I show you another one. Yes. So this is kind of image and update the figure. You can see resolution here, still here. FSC, this go down, never go up. This is between map. This is between map model. And they all go down without go up. All data like this.
Okay.
Hope that convinces you.
Yes.
Yeah, good question. So review ask the same question. We update the figure. Sure.
Okay, so anyone else want to ask anything that they have in mind? Online, I think online audience.
Yes, I said, I have a question.
Of course.
Yes. So, like you have talked about conformation of the proteins, right? And you
Yes.
have used cryo-EM, which means you have to freeze the proteins in a liquid nitrogen, right? So, how do you
Yes.
ensure that that freezing part doesn't change the conformation?
This is pretty good question. So first of all, this is the kind of traditional people do. And they first they frozen in a liquid acid instead of liquid nitrogen. They keep sample in liquid acid. So the frozen speed is very fast and less than can they calculate a couple of milliseconds. And the speed is so fast, not even the ice have no chance to form a crystal. That's why it called amorphous ice. So people use that one to frozen cell, frozen even like animal or something, living cell. And they found that all cell functions still there. They think if I frozen a protein, and protein must be native conformation. So this is the guessing, and no one actually tell. This is exactly no conformation or not. And general people accept that if I frozen cells work, still living cell and a Y protein and change. So this is kind of traditional people use it. No one kind of question about this one, but it's good. No for sure answer.
Okay, I have one more question. Like using electron microscopy, is it possible to distinguish right-handed helix and left-handed helix, which are the secondary structures of proteins?
Sorry, I didn't know what. I didn't catch what.
Yeah, so like proteins will have structures like helix, no alpha helix.
No alpha helix. Possible. And as long as we still challenge the resolution. Right now we're pretty good on the DNA RNA structure. And the lucky is DNA RNA is a big density. Helix is 2 nanometer, and we barely just see the helix of the DNA RNA. But the protein helix much smaller. This is the 8 angstrom. And we still need to optimize our condition and then you want to get a protein helix structure. So we haven't go that far. And but for RNA helix or RNA loop, so we pretty easy distinguish them. And here you see the bridge between the density, they are much skinny and less density. This is the helix bridge part and the loop part connection together. So yeah, it's still a long trip to go high resolution, but the good thing is we don't choose in the sample. That's why we gave the wide distribution, give you all picture, but the client or other method only narrow down one or two conformation structure, and they go much higher resolution. So different strategy to see the molecule.
Oh, thank you.
Yeah, thank you. So, Gary, I have a couple of questions that I thought if you can comment on. Can you please comment on the temporal resolution and the noise that we see in the images, how to reduce it or how to tackle these noises?
One is time resolution or something. I didn't get your first one. Time resolution you are mentioned.
Yes. I was thinking that one day, I mean, what are these systems are capable of? Like, can we take images continuously, as the things move on? So what could be the temporal resolution or something like that?
Yeah. Okay. So let's see, and hopefully I can show the background here. So this is kind of dynamic process, take like 10 hours. Lucky the sample take 10 hours. Too short time, it won't be able to catch it. And for this is the extreme case, and we choosing sample is 6 hours during the process of the maturation. So in this 6 hours give us a distribution versus conformation. It's pretty wide range. We also can frozen sample in 3 hours or 10 hours or 0 hours. And still maybe see the conformation difference and the variation. So we haven't do it, but we plan to repeat this one at different time. Hopefully the distribution and of the molecular gave you something about dynamic process with time. So just like we think that in a chemical reaction process, the molecule is go different pathway. That's why the process is quite different each other. Just like a example, in people in the meeting room, you suddenly tell them, let's go lunchroom. So I believe somebody go very fast. They go like 2 minutes, 3 minutes later, somebody already in lunchroom. And somebody is there talking other and in the meeting room. Many people in the pathway. And if you're frozen and the people in the process, you find some of them already in the meeting room, some is in the classroom, a lot in the middle. Based on comparison, tracking position, their location, you barely gave them about the folding process. And by different time, you can see how fast they're going. So this is another way and study the conformation difference and based on time. And you get what's the distribution different between early stage and late stage instead of choosing like single party and choosing of them. If you're choosing them, maybe you're always choosing the same high population one, won't be able to see the difference between particles. And or some is a continual change, may never get any high population conformation. So this is kind of compare the time resolution TEM and the benefit out here. But again, without average resolution much lower, so it's still very challenging. So the major noise from the background imaging, and this is the ice here, buffer here, and ice thickness is a big gain. That's why, and we optimize condition including this ice thickness issue. And thinner ice, you get a much better transparent signal noise ratio. And when you collect data, definitely you try to use the worst highest dose you can use. You can intend damage a sample to see where's the limitation for your resolution. And if you target like a 1 Armstrong or 2 Armstrong resolution, the dose limit is much lower. If you target like a 20 Armstrong, and you can have a higher dose. So this is the kind of target resolution, also depend on dose issue. Of course, dose rate as well. Those kind of way can help you reduce the noise level from a data collection point of view. And of course, from an image process point of view, there are a lot of things you can do to recognize what's the noise level and by the image process in a way and like a machine learning to figure out what's the noise level and some filter in to the some resolution you target. You ignore, like say high resolution, you don't care. You just report the high resolution part and increase your contrast. So tons of the method you can strategy can help to increase the image contrast and by reduce noise.
Yeah, yeah, we are almost close to the end. I have a request that I see that one of your paper, there are a lot of work that you've taken, you have talked about. This paper is in print, I guess, in Nature Communications. So can you, once it is published, can you please send us the link so that we can tag with your talk so that it,
Okay.
people can access it.
Of course. Thank you very
Yeah.
much for sharing.
Yeah.
So, and once it's the printout also, we definitely will send you and share this study.
Yes. Okay, so if no other questions, then we can just thank the speaker for such a great talk.
Yeah. Thank you very much.
Yeah, thank you,
Yeah.
Ren. Thank you.
And
Thanks a
so
lot.
nice talk with you.
Yeah,
Scientists
see you
and
soon.
students.
See you soon. Yeah.
Yeah. Bye.
Bye.